Hello, in this video we'll be taking our first look at enzymes. So let's not waste any of your time, let's get right to it. Now when we're talking about enzymes, we're talking about the player that essentially is doing all the work that we've been talking about. When we think of biological catalyst, when we're talking about hydrolysis and dehydration synthesis and anabolic pathways and metabolic pathways and endergonic reactions and exergonic reactions, the one thing that we saved for this is the idea that enzymes are the ones that are doing all of these work. All the work that we've been talking about. So we're going to connect enzymes to all that stuff. Now we also want to recognize that most enzymes are proteins. There are a few that are RNA-based, ribozymes, because of the ribose sugar. But it's the idea that these proteins, these enzymes, are essentially, we're going to look at a bunch of examples here, and then when we do our in-class activities we'll see them as well. But it's the idea that these enzymes, for one, it's all a shape-driven recognition system, just like your lock, your key in your lock. Only particular keys can open particular locks because they have to match up the exact shape, and enzymes and substrates molecules are the same way. The enzyme is designed for a particular shape, and only molecules that it's designed for will fit that shape. So the enzyme that is meant to break down lactose could not do any work on glucose. The enzyme is designed to separate the 6-carbon strand of glucose in glycolysis, could not do anything with lactose, and vice versa. They are specifically designed to do the one job that they're designed for, and because simply the molecules are different shapes and they wouldn't fit the shape. So it's the idea of an enzyme. You can see in that diagram, it's got what is called an active site, and that's where the molecule or the substrate will position itself. And once that happens, then the enzyme can work on it. And really the work that's being done, it's either creating bonds as it builds molecules, or it's breaking apart bonds as it breaks apart molecules. So really the work the enzyme is doing is on the bonds themselves, either creating them and building, or breaking them and breaking down molecules. Now, one of the things, along with working on the bonds, that molecules or that enzymes do is to make the reaction happen quicker. Happen quicker, and it does so by altering the needs of the reaction. And when I say that, what I simply mean is that it lowers the amount of energy that is needed to start the reaction. So if you think about a couple of good examples, if we're going to lower the amount of energy needed to make the reaction, that could be similar to, or thinking about lowering the boiling point of a metal. If I lowered that boiling point, that means it's going to boil at an earlier temperature. At an earlier temperature. If I lower the boiling point of say lead, say I lowered lead to a boiling point of 60 degrees. Okay, that would change how we could use lead to build or to make tools. So if my hammer, I got in my shed, I actually have 3 hammers. None of them seem to hit the nail right, but that's a whole different subject. But the heads of the hammer, or the part that used to strike the nail, or also the claw end to take the nails out that I bent, are all made out of lead. So what would happen to my hammers if the boiling point of lead was less than 7 degrees? Well, you guessed it. It would have to stay inside the house, and the house be kept above 7 degrees, because if it got below 70 degrees, then the lead would just, it would start to boil. And I said it backwards. It would have to be kept in the refrigerator, not just in the house to keep warm, but it had to be kept cold, because if it got above 60 degrees, then it would start to boil and evaporate. And you don't want your hammers to evaporate. So that's the idea of lowering the energy level, and that speeds up what's happening. If I lowered the boiling point of lead, it would get to boiling point quicker. Another way of thinking about this, if you're obviously if you're a Nevada high school student, you're familiar with Nevada. If you're a Crowder student, maybe you are familiar with Nevada, maybe you're not. But over on Atlantic, if you want to think about enzymes and how they help speed up the reaction, Atlantic, going from ash to olive. You're headed from ash and you're headed to the middle school, or maybe you're going to the dirt bike race track out at the edge of Atlantic. But at the western end of Atlantic, there is a very steep and long hill. Okay, very steep and long. So if you were headed west on Atlantic on your bike, okay, and you got to that hill, it would take you a long time to get to the top of the hill, and someone like me eventually would just stop and walk the bike up the rest of the way, because it just wasn't going to happen. Now, if an enzyme was going to come along and help in this situation, it would lower the grade of the hill. In other words, it would just put its enzyme hand, for lack of a better term, on the top of the hill and push it down to where the steepness wasn't as much, or the length nearly as long. And that would speed up the time it would take to get to the top of the hill. So enzymes do the same thing. They reduce the amount of energy needed, and lower the activation energy. You can see on the graph, the original amount of energy needed is the purple line, but when an enzyme engages in the reaction, it lowers it down to the green line, so it can happen quicker. So in this picture here, we're going to identify some sites. We've already mentioned the active site. That is simply where the substrate attaches. So in this diagram, the 2 little blue circles where the substrate is going to set into, that's the active site. In this case, you can see that it's a catabolic pathway, because the end result is to break apart the molecule. And again, the enzyme could work on the individual parts, but for the most part it works on the bond. So I take this disaccharide perhaps and make it into 2 monosaccharides, and the whole the whole thing, once the substrate is in there, that is called the enzyme substrate complex. Simply just reference to everything that's involved at a particular time. So the substrate is the molecule. The place where the molecule attaches is the active site. When the substrate is inside of the enzyme being worked on, that's called the enzyme substrate complex. And of course, once it's done, that's the product. In this case, you have two monomers now. Now, the idea of altering those bonds, it does it with pressure. And that pressure that it applies to the substrate is called induced fit. Just simply a reference to the work that's being done on the substrate. Okay, here's another picture to show what we've been talking about. The enzyme obviously is the large blue thing, and the substrate is a molecule that's about to be worked on, and so it sets into the active site, and you can see it kind of closing around it. So that's the pressure, that's the induced fit that we just talked about, and whatever the enzyme is going to do to the molecule, it is now doing. All right, so in another example of what we've been talking about. Now here again, this breaking apart, you can see those end products, and not all enzymes are just breaking apart. We build at times as well, and when we get in class, you'll see a simulation that show. It's actually a game that shows that. But in the induced fit model, or the idea of that pressure on the substrate, both the substrate and the enzymes change shape. Just like if you were giving someone a bear hug, you would change shape, they would change shape. That's enough on that. So the enzyme puts the pressure on the substrate, and therefore changes it. And when that enzyme is putting that pressure, that's going to change the rate of reaction. In other words, change how quickly it can happen. And in the example here beside me in the diagram, you can see that not only do we break apart the molecules, but we are also changing the individual components. You can think of salt in that manner. When they've got salt made, it's a particular shape and structure, but when I separate the sodium and I separate the chlorine, they change into entirely different items. The product is different than what the substrate was. Okay, so here's just another example. You can see that I've got my disaccharide or my molecule. Okay, the enzyme, again, it sets into the active site. Okay, again, we're breaking apart. So this one shows that water coming in. So we're talking hydrolysis to get that to happen. And of course, the final product is now they're separated. And we've broken apart the molecule and connecting it to that breaking apart process. Now, some functions, okay, of enzymes, and we're going to introduce some new angle here, and in video 2, part 2, introduces a bunch of different new angles or things that affect the performance of an enzyme. But I mentioned that each molecule has its own particular enzyme that's going to fit its shape. That's true in theory, but some molecules don't have an actual full enzyme assigned to them. Or some molecules have what they have what they call coenzymes. In other words, it's the shape of them which will allow an enzyme to be converted to that shape. So you can see here that I've got an enzyme, I've got an active site on the far left, and it works on a particular molecule. That's its primary job. But every once in a while it may be needed to work on a different molecule that isn't it's the traditional one, and a coenzyme can be used to alter the enzyme into the shape of the second molecule. You can think of it as an adapter. Okay, we have an older house, and a lot of you buy equipment or things today, and things today typically have the 3-prong outlets. All our outlets are 2. So to make it fit, you've got to buy one of those 3-prong to 2-prong converters, those little gray things, and then you can plug things into the outlet. This is the same idea. The coenzyme is the shape of the second molecule, and it just sets into the active site of the enzyme, and then the second molecule now fits. And when it's done working on that molecule, the coenzyme can come out, and the enzyme goes back to its original shape. We can also think there's also cofactors, factors that can influence the speed of the reaction. But something else to think about, and we'll wrap up the second video with this concept, is that a lot of times concentration amounts determine the speed of the reaction. If an enzyme has a bunch of work to do, then it does that work quickly, individual tasks quickly. If it doesn't have much to do, then it does the individual acts at a much slower rate. Well, that was our first look at enzymes, and I know that was a bunch, but when we come back the next time, we've taken our second look at the things that influence or affect enzymes and their work. So we'll see you then.